My name is Jessica Durston. I'm going to be speaking to you today about organoids, specifically what they are, how they're being used in research, how they are cultured, what are the advantages and limitations of using them as a model. So, what are organoids? They are tiny self-organized tissue cultures grown within a three-dimensional extracellular matrix. They're used for studying human and animal tissues because they resemble whole organs in structure and function. They are used in research in a variety of ways, as you can see in the diagram on the right. Applications such as studying mechanisms of the disease, generating knowledge and tools applicable to preclinical studies, to study infectious diseases, genetic disorders, and cancers. Organoids can be generated from any individual, allowing the study of variability among individuals at the tissue level, as well as the cellular mechanisms leading to complex disease phenotypes. Organoids can be derived in different ways. They can come from pluripotent stem cells, including embryonic stem cells or induced pluripotent stem cells, or from tissue-derived stem cells of a given tissue. Organoids are amenable to many standard in vitro assays, including RNA and DNA isolation, immunohistochemistry, and genetic manipulation. Organoids can be initiated from cryopreserved material, then expanded, and then cryopreserved for future use. For the culture protocol, the figure on the bottom is a basic outline of organoid culturing. There are many resources online which list all the actual steps and necessary reagents, including an organoid culture guide from ATCC, from which this figure was borrowed. Individual cells or organoid fragments are embedded within a liquid extracellular matrix, or ECM, and dispensed as small droplets onto the surface of a warm tissue culture plastic vessel. The ECM will solidify into a gel after incubation at 37 degrees C, and then it will be covered with the culture medium. Organoids will develop within the dome as 3D structures. The figure on the top right is an example of a workflow of the culturing process of urine-derived bladder cancer organoids. The paper from which this figure was obtained was looking at whether it would be possible to establish tumor organoids from the urine of bladder cancer patients as an alternative to tissue-derived organoids. Organoids should appear in culture within 3 to 7 days as small ring-like or spherical structures with smooth, defined edges. At 7 to 14 days, larger, clearly visible organoids should be apparent. Model-to-model morphology can vary significantly, with some being small and solid pebble-like organoids and others being large and balloon-like. The 4 black and white pictures on the right are an example of an organoid at different points in time and development. For relevance and controls, the organoid culture is relevant to what we learned in Module 5, which was culture and manipulation of eukaryotic cells. It is basically tissue culture. It is special due to the tissues that are being cultured. For controls, organoid culture is usually done manually, mostly using traditional cell culture techniques. Controls for this technique include using the correct media for each particular organoid. This may be a process of trial and error if establishing a new one. If you can send the organoid samples out for pathogen testing if you are purchasing them rather than creating them in-house. Avoid microbial contamination by using good tissue culture techniques. Perform procedures in a biosafety cabinet. Make sure that the extracellular matrix is not contaminated. If possible, send that out for pathogen testing as well. The figure at the bottom is from a paper looking at synthetic alternatives to Matrigel. Matrigel is a commercially available extracellular matrix. This figure gives you an idea of how the extracellular matrix supports cells and organoids in general. The advantages of this technique versus other techniques and models. Other models that would be comparable to organoids are in vivo models or 2D cell cultures. Organoids offer an ethical advantages over traditional in vivo models as they can replace or reduce the need for animal testing. Organoids are better than 2D cell culture because they simulate complex interactions within organs, which doesn't happen when all of the cells are the same. Organoids offer faster results when compared to using whole animals. Organoids can be patient-specific, and the figure on the right is from a paper which generated immunofluorescence images of functional and structural proteins and physiological functions of the liver in hepatocyte organoids. This figure shows the complexity of protein interactions within hepatocyte organoids. Some of the limitations of this technique include cost. Buying organoids is expensive. The catalog entry on the right-hand side shows that one sample is over $3,000. It is not appropriate for all tissues because organoids lack in vivo complexity of some tissues, for example, skin. The organoids obviously lack an immune system as well, and that is usually very important to the study of disease. Thank you for taking the time to listen to me, and good luck on the final.